Hey lab experts, welcome to this explainer. Today, we're going to build and really refine those rapid visual identification skills for nematode eggs using actual direct clinical data. If you're spending your days hunched over a microscope, you already know that speed and accuracy are literally everything. So let's sharpen both, shall we?
So here's the big question. How do you instantly and accurately differentiate similar nematode eggs under the glass? Let's dive right into this, because differentiating these microscopic lookalikes is often the absolute toughest part of parasitology diagnostics. You're dealing with minute size variations, super subtle differences in shell texture, and internal structures that, let's be honest, can easily blur together if you aren't sure exactly what to target. Well, we're going to completely eliminate that guesswork today.
All right, here's our roadmap for today's session. First, nematode egg morphology basics. Then, distinctive shapes and larvae. Third, differentiating polar plug eggs. Fourth, comparing blastomere egg samples. And finally, diagnostic reference tables.
Let's jump right into Section 1, Nematode Egg Morphology Basics.
Now, before we start comparing those tricky samples, we need to get our fundamentals straight. The crucial takeaway here is that we aren't just looking at the overall shape. I mean, sure, knowing if an egg is oval or barrel-shaped is a great start, but true diagnostic precision, that requires you to identify very specific structural components. We're talking about rigorously checking the blastomere count, confirming the exact direction of polar plugs, measuring that shell thickness, and noting whether or not any actual internal larvae are hanging out in there.
Okay, moving right along to Section 2, Distinctive Shapes and Larvae.
We're going to kick things off with two nematode species that really stand out from the crowd, thanks to some highly unique morphological traits. First up is Toxocara. What you're seeing here is a classic clinical sketch, and it just perfectly highlights the textbook hallmarks of this species. Notice that highly distinctive, thick shell? More importantly, take a close look at the heavily pitted surface wrapping around those dense granular contents right in the center. Seriously? Burning this mental sketch into your brain is your absolute first line of defense at the bench.
Now, it's always super fascinating to see exactly how that idealized sketch translates to real-world clinical work under the scope. Look at that. You can distinctly see that thick, dark shell practically jumping out from the background. And that pitted texture we talked about, it gives the egg a very robust, almost rough appearance, especially when you compare it to smoother nematode eggs.
So, let's quickly synthesize your rapid diagnostic checklist for Toxocara species. Basically, you're hunting for an egg that is definitively round or spherical, and notably dark in color. It should measure right around 75 micrometers. If you spot those granular contents packed inside that unmistakable thick, pitted shell, boom, you've got your positive ID.
All right, shifting gears to strongyloids. This side-by-side view is great because it perfectly bridges that gap between textbook theory and clinical practice. On the left, the sketch really emphasizes a clear oval shape and a distinct internal structure. But on the right, the actual clinical view shows us a relatively pale egg containing a very well-defined, fully formed larva just curled up inside.
Honestly, the absolute key diagnostic feature here is finding that grayish S-shaped larva. When you spot an oval, pale, embryonated egg measuring exactly 65 by 25 micrometers, your eyes should immediately dart straight to the center. Look for that curled larva. If it's staring back at you, strongyloidsia is your diagnosis, for sure.
Let's roll into Section 3, Differentiating Polar Plug Eggs.
Okay, we are now tackling one of the most common, frankly annoying, diagnostic mix-ups at the bench. By placing Trichuris right next to Capillaria, we're basically mimicking your exact mental process at the microscope. You scan a specimen, see an egg with plugs at both ends, and your brain instantly has to rule out possibilities based on really subtle structural differences. I mean, both have plugs, both are brownish, but you've got to look incredibly closely at the shape of those plugs and the overall contour of the shell.
And this right here highlights the exact contrast you really need to commit to memory. Your definitive diagnosis hinges completely on those plugs and your micrometer reading. Trichuris features extruded polar plugs, meaning they push outwards quite prominently, and it measures a larger 80 micrometers. Capillaria, on the other hand, is quite different. It has intruded polar plugs that sit much flatter against the shell, and it comes in way smaller at just 50 micrometers.
Let's break down Trichuris a bit more. You are specifically confirming a very distinct lemon or barrel shape. It's got a prominent brown color, and when you measure it, you're looking for dimensions of 80 micrometers by 42 micrometers. And of course, it's capped by those unmistakable extruded polar plugs that literally stick out like tiny little corks.
Conversely, when you're looking at capillaria, the shape is much more of a standard oval, not a barrel. It still has that brown coloration, but those polar plugs are notably intruded. They definitely don't protrude outward abruptly like the last ones. And critically, your reticle will confirm a smaller total length of just 50 micrometers. Up next, Section 4. Comparing blastomer egg samples.
Welcome to what I like to call the blastomere battle. Looking at Hamanchos contortus and hookworm side by side, the challenge is pretty obvious, right? Both of them present as light brown ovals. Both feature very thin, really smooth shells. To the untrained eye, or honestly even during a rushed visual scan by an expert, they are incredibly easy to mix up. You absolutely cannot rely on shape and color alone here.
So here is your diagnostic tipping point. When you are faced with this thin-shelled oval, you simply must take a breath and patiently count the blastomeres inside that delicate yolk membrane. Haemonchus contortus is going to have more than 8 blastomeres, while a hookworm egg will have somewhere between 4 and 8. Plus, you can verify your count with a quick measurement. Haemonchus is slightly longer at 86 micrometers compared to the hookworm coming in at 74.
Let's summarize. For a definitive Haemonchus contortus call, lock these traits in. Oval shape, thin smooth shell, light brown. But the absolute deciding factors? Measuring that 86 micrometer length and confirming strictly more than 8 blastomeres packed away within that delicate internal yolk membrane. Now hookworm, it totally shares the oval shape, the thin smooth shell, the light brown color, and that delicate yolk membrane, but the game changer is that it will consistently contain a countable 4 to 8 blastomeres, no more than that. And it measures slightly smaller in your field of view, at exactly 74 micrometers in length.
All right, final stretch. Section 5. Diagnostic reference tables.
To really cement all this data for your daily lab work, this table right here perfectly synthesizes all the critical structural data we've just covered. It neatly aligns the species, the shape, the precise micrometer sizes, and most importantly, that one golden key distinguishing feature for every single one. From Toxicara's pitted shell and Strongyloid's S-shaped larva, to the great plug debate between Trichuris and Capillaria, and finally the blastomere countdown between Haemonchus and hookworm. Taking a quick mental snapshot of this matrix provides a really rapid, foolproof visual identification guide for your incoming clinical samples.
So, armed with this highly specific comparative data, a lot of the uncertainty at the microscope simply vanishes. I'll leave you to ponder this. The next time a really challenging specimen crosses your desk, which of these subtle diagnostic details will be the master key to cracking your next microscopic mystery? Thanks so much for joining me on this explainer today. Keep those lenses sharp, and we'll see you next time.